What are the tools of hematopathology? This was the challenge from yesterday. What did you find? How did you organize them? Here is my way of organizing the top 10 tools of hematopathology. I've summarized them by analysis target and analysis type. If we look at the targets, there are several targets we can consider. The whole hematopoietic system, cell morphology, protein markers, chromosomes or entire genes, parts of genes or base pairs, going from large to small. Different analysis types can be single or multiple. If we consider the whole hematopoietic system, a snapshot that we can acquire is the CBC, the complete blood count. From that, we can tell if there are increased lymphocytes, increased blasts, the number of other white blood cells like neutrophils, platelets, and red blood cells. All of these are important in coming up with a hematologic diagnosis. If we look at all of these features over time, we construct the clinical history, where we can look at trends within the white blood cell count to see if there is a persistent anemia, for example, or a persistent leukocytosis, or a persistent thrombocytosis. All of these are important for landing upon a diagnosis. The second row, cell morphology. We can look at this from a single cell perspective, for which we use the general term cytology. So the size of cells, the cytoplasm, the nucleus, whether there is any nucleoli, the texture of the chromatin. And if we look at multiple cells together, we will call that architecture or the arrangement of cells. Are the cells in question clustering together, forming diffuse sheets, or are they scattered? Moving down, we can look at the protein markers within the cell or on the cell. If we do it one at a time, we'll call that immunohistochemistry, where we can use different markers such as CD20, CD3, CD10 to look at one cell and one marker. If we look at multiple markers at the same time, technique that we can use is flow cytometry, where we look at multiple markers within the same cell. And we look at multiple markers with multiple cells. If we dig deeper into the nuclear material, we can identify chromosomes and entire genes with fluorescence in situ hybridization, or FISH. With this approach, we can identify one gene at a time and see whether it is intact or whether it's broken apart into smaller pieces and gets split up and sent to another part of the genome. If we look at multiple chromosomes, the technique is called a karyotype, where we'll display the entire genetic material by chromosome number 1 through 23, and then X and Y. Or 1 through 22, and then X and Y. Going even smaller, we can look at parts of genes or base pairs. We do it one at a time. We can use the tool of PCR, polymerase chain reaction, to analyze a target base pair within a target gene. And if we look at multiple genes or multiple base pairs within the DNA or RNA, we can use the tool of next generation sequencing, or NGS. All these together are the top 10 tools of hematopathology. Other areas in pathology, such as surgical pathology, may have other tools that they use, such as um, um, uh, methylation of DNA. However, within hematopathology, that is not one of our major tools that we use yet. If we look at these top 10 tools, we can actually compare them to the initial 3 characteristics that I mentioned before: doing bad things, abnormal phenotype, and clonality. And we can see how each row can correlate to one of the features. CBC and history, that helps us prove that a process is doing bad things within the patient. Cytology and architecture can also prove that it's doing bad things if it's taking over a place where it should not be taking over. Now, abnormal phenotype, we can see with immunohistochemistry or flow cytometry. And clonality, we can prove by showing different rearrangements of chromosomes or genes using FISH or karyotype, or even looking at parts of genes with PCR or next generation sequencing. With these tools are the transportation that we use to navigate throughout the Hemepath world and will help you go from one place to another in your diagnostic journey.